One.
Okay. Hi. So my name's Megan, and my other partners were Jessica and Amani, and we did the comparison of differentially expressed genes in Alzheimer's patients and mouse models using RNA sequence. So just like a little background information on Alzheimer's disease, it's a deliberating neurological disease that affects memory and other mental functions. The brain cells basically degrade and die, causing symptoms such as behavioral problems, language issues, and memory loss. It's caused by an increase of proteins in and around the brain cells, which I thought was pretty interesting actually with like the results later when we'll talk about it. It also affects a big portion of the population every year, and a number just keeps continuing to grow. Um, a major issue with this disease is that there is no cure for it, and there are drugs that can help combat like some of the symptoms, but the progression, um, ultimately, there's no cure for it, I guess. Sorry. So our research question was focusing on changes within the brain and how does the RNA sequence show the differences of a healthy patient and a patient with Alzheimer's disease, and also looking at like mouse models too.
So for the methods and materials, these are just the papers that we used and what they used to figure out what the Alzheimer's genes were. They used gains and GWAS data, as well as RNA-seq and snRNA-seq. Methods, materials, GEO sequence were pulled from various articles containing RNA-seq of healthy, those with Alzheimer's disease, and also mice that were healthy, and then some that had symptoms of Alzheimer's disease. Those SRR sequences were gathered from the NCBI database and were put into a genome generator known as cyverse to collect our sequences. So for Homosapiens, there were 5 SRR sequences that were gathered, and 3 that were unhealthy with Alzheimer's disease, and 2 being healthy without Alzheimer's disease. And for the mouse or mus musculus, 6 SRR sequences were gathered, 4 being unhealthy with Alzheimer's disease and 2 being healthy without Alzheimer's disease. And then once our sequences were gathered, we were able to plug them into RStudio to collect data for our volcano plot.
So for the results for the Homosapiens, basically the downregulated genes, we did 10 of them, and we did 5 that had a p-value of 0, and then a p-value that was greater than that. Something happened with our information, but after examining about 10 of those downregulated genes, we noticed that each of them possessed a similar change in the way that proteins are being encoded to the different cells. And there was an interesting gene that was linked to Calmodulin family. Its mutations in this gene have been identified with causing epilepsy and delayed neurodevelopment. It seemed that most of them, the downregulated genes, were related to a delayed neurodevelopment too. And then for our upregulated genes for the Homosapien, we did 10 also for that one too. And we saw that they expressed more neurological changes that were like mutations. A couple interesting ones was that there was a potent neurite outgrowth inhibitor, and this blocks the regeneration of central nervous system in higher vertebrates. And then the other one, it causes axonal neuropathy, which they're abnormally large and dysfunctional axons, and they can cause issues with nerve impulses, which I feel like makes a lot of sense with Alzheimer's disease.
So for the mus musculus, the downregulated ones, the gene, this gene right here, or copine-7, is a membrane-binding protein that is calcium-dependent that may regulate what occurs at the interface of the cytoplasm as well as the cell membrane. And then this gene, or MYAT, is a gene that encodes a long non-coding RNA that may be a component of the nuclear matrix. And then for upregulated genes, this gene, or high mobility group box 1, is a gene that encodes for a protein that is responsible for regulating transcription and how DNA is organized. And then this one, period circadian clock 2, that is the primary circadian pacemaker in the human brain. It encodes for locomotor activity, behavior, and metabolism.
And then just for our discussion, the results of the volcano plot shows that there's some similarities between the mice and the humans. This can be really helpful with further research too, and trying to find more information about Alzheimer's disease. The most common traits that we saw with these genomes were mutations of the genes, and they would cause neurological disruptions on the brain. Some common things that we noted were locomotive and behavior changes, changes in neurotransmitters that were shortened or stopped signals from being sent to various parts of the brain, mutations of different genes that caused a change in the way that proteins were attaching to different cells. And a lot of them actually showed different tumor suppressor genes, which were also common in a lot of them that we looked at. Mostly common, I think, in the downregulated ones, actually. But yeah, that's it. That is our presentation.